Then I am Shashidharamurthy. I am chairing this session. First of all, I welcome you all. And I thank OMICS groups for giving us an opportunity to share our investigations herein. And having said this, today I will be talking about a protein, a innate immune protein. That means the first line of protein which get upregulated after any injury or stress. That's what, but its role in autoimmune disease is still obscure. So today I will going to share some of the findings, what we found and what is its role during autoimmune disorders, okay, inflammatory disorders. First, having said that, to go to the background of the study, lipocalin-2 is a small protein which is known to be involved in iron transportation. That means what they do is they take the iron from the extracellular space and then they put them inside or transport them inside the cells for the survival. And it is, as I already said, it's a small protein with a molecular weight of 20 kilodalton, and it is first identified in neutrophils. And what's its role? There are several roles. So far, the characterized function of the lipocalin-2 is the antibacterial activity. So what is antibacterial? What it is doing with the antibacterial? How it is doing the antibacterial? What it does is it secretes and chelates the iron from bacteria so that bacteria cannot survive. That's the main function of this protein, and it is well characterized. Apart from this, recent findings are also shown that they are also involved in cell proliferation and migration and during the development of cancer and the tissue repair, another mechanism. How? It is helping in the tissue remodeling process. The good example for this is after the delivery, the ovary. To regain the normal size of the ovaries and uterus, this lipocalin is highly required. So that, suggesting that it is required for tissue remodeling process. And it is also known to be involved in during apoptosis process. Okay. Having said this, during autoimmune disease, in most of the autoimmune disease, it gets upregulated. So what is the function? Why it is getting upregulated? It's still not known. So our intention is to see what is the functional role of this autoimmune diseases. Functional role of lipocalin-2 during autoimmune disease. That's the main focus of my talk. So as I already said, lipocalin-2 is involved in infection, inflammation, injury, and stress. During these conditions, it gets upregulated. At mRNA level, it almost goes up to 1,000-fold increase at mRNA level, whereas in the protein level, it almost goes from 10-fold to 500-fold depending upon the condition. Okay. Apart from that, the cytokines, the pro-inflammatory cytokines, and the leptin, retinoic acid, and TLR ligands such as LPS, poly-IC, they are all known to induce the lipocalin-2. What are the cell types what are the cells which are involved in the secretion of lipocalin-2? Mainly, it is ubiquitous. Most of the cells secretes the lipocalin-2. But for the purpose of our talk, we will be focusing on neutrophils and macrophages because these two cells are very important. They play a very important and vital role during the pathogenesis of autoimmune diseases. So our first question is Lcn2 involved in antigen-antibody complex mediated inflammation? What it means is during autoimmune disease conditions, autoantibodies will be produced and they bind to the target antigens and forms a complex that we call it as antigen-antibody complex or the immune complex. So our question is whether this immune complex induces the expression or upregulation of lipocalin-2. To check this, we set up one experiment. So what we did is we took the macrophages cells and then incubated with the immune complex. After at 37 degrees centigrade for 24 hours. Then we took the culture supernatant and then assayed for expression of lipocalin-2. As you see in this, when we incubated the cells with or macrophages cells with immune complex, the Lcn2 get upregulated, significant upregulation. This data suggests that immune complex induces the upregulation of Lcn2 through what pathway? It is through Fc gamma receptor mediated pathway. So during autoimmune disease conditions, Fc gamma receptors are the one which play a major role. So this experiment tells us that the Fc gamma receptor mediated upregulation, the Lcn2 upregulation is mediated through Fc gamma receptors. Okay, now this is an in vitro experiment. Now we wanted to check out, is it true in the in vivo condition? To assess this, to answer this question, we carried out one experiment. It is called Arthur's reaction. Simply, we can call it as a skin inflammation model, antibody-mediated skin inflammation model, and also it's an example, a good model to study the anaphylaxis, IgG-mediated anaphylaxis, the allergic reaction. Okay, so in this, what we do is we take the mice and we inject the antibody at a specific site, on the specific site intradermally, and then the antigen along with the blue dye, we injected IV intravenously, so through tail vein. So what happens after 3 hours, the antigen antibody makes the complex and it induces or initiates the inflammation. During the initiation of inflammation, there will be a vascular damage, right? So during this vascular damage, along with the antigens, the blue dye, what we injected, also diffuses into the site of antibody, the antibody injected site. That means wherever the inflammation takes place, this blue dye also leaks in there. So the intensity of the blue dye is directly proportional to the intensity of the inflammation. That's the main point which I'm trying to explain here. Okay? So what we do is we measure the intensity of the blue dye and we correlate with what is the intensity of the inflammation. So when we did this, so to answer what is the role of lipocalin-2 during this process, the antigen antibody mediated or immune complex mediated inflammation, we took the advantage of a lipocalin-2 knockout mice. So we repeated the same experiment with lipocalin-2 knockout mice, and then we observed for the blue dye extravasation. So as you see here, this is the wild type mice and this is the knockout mice. So when you observe the blue dye, it's very evident that the inflammation, a reduced inflammation in this mice. Okay? So the inflammation is reduced, suggesting that lipocalin-2 may be required for the initiation of inflammation, antigen antibody mediated inflammation or immune complex mediated inflammation. So that's the first clue we got. And these two, the 2.4G2, 2.4G2. This is a specific antibody. This serves as a specificity control for. So what does it mean? It is a monoclonal antibody which neutralizes the antigen antibody mediated inflammation. So as you see in this picture, you don't see the inflammation, suggesting that this antibody completely inhibits the immune complex mediated inflammation. So the take-home message from this slide is the lipocalin-2 is required for the initiation of inflammation during immune complex mediated inflammation. So then what is it? What's happening in the inflammation site? To answer this, we took the samples from the sites where we injected the immune complex, and then histopathological studies were carried out. So as you see in this, the previous injected site, the first as a control and non-inflammatory. Okay, this is lipocalin-2. Here we are comparing wild type and non. So if you observe the antibody injected or the inflammatory sites in the wild type mice, there is a lot of infiltration of inflammatory cells. Whereas the infiltration of inflammatory cells is significantly reduced in lipocalin-2 knockout. That means So now, to substantiate our result, what we did is we administered the recombinant lipocalin-2 to the lipocalin knockout. That means we exogenously administered the recombinant lipocalin-2 to the knockout. So then, after an hour, we carried out arthritis skin inflammation. We induced the skin inflammation. And what we observed is, after the administration of recombinant Lcn2, the knockout mice exhibited almost similar inflammation. The intensity of the inflammation is almost comparable with the white type knock, suggesting that the lipocalin-2 strengthens our point, saying that lipocalin-2 is required for initiation of inflammation. So next, to further substantiate, what we did is we neutralized the naturally secreting lipocalin-2 or Lcn2 in white type. So when we neutralized it by using a monoclonal antibody specific to the lipocalin-2, what we observed is in the white type mice, this is the treated mice with the Lcn2 monoclonal antibody, the intensity of the inflammation is significantly reduced. If you look at this bar graph, you can easily measure. See, this is the Lcn2 monoclonal antibody treated white type mice. If you compare the bar graph, it is significantly reduced the inflammation. Intensity of the blue line, in turn, it significantly reduced the inflammation. Okay, it is neutralizing the function of circulating Lcn2. Okay, next slide. Let's move on and see. Now, this arthritis reaction or skin inflammation model, what I explained now, is only for 3 hours. It's a very acute condition. Now, to further, we wanted to investigate whether this is true in terms of autoimmune disease conditions such as arthritis. That is longer, right? It's a persistent inflammation, a sterile and a persistent inflammation, and stays for longer period of time. Now, we wanted to investigate whether the same function is true with respect to autoimmune disease conditions. To do this, to answer this question, we selected a model called serum-induced arthritis model. Here in this, model, what happens is that it develops the autoimmune arthritis. Now, the beauty is, if you take the serum from this mice and inject into any mice, they develop arthritis. So that's what we did. So this slide just explains how these mice develop the autoimmune arthritis. Now, what we did is, this is not the model what we used. What we did is, we took the serum from that progeny, F1 generation, and then injected into our mice. That means lipocalin-2 knockout mice. What we did is, we took the serum from this mice and injected intraperitoneally, and then we observed for the pathogenesis of arthritis in lipocalin-2 and wild type mice. And then we scored the disease. So what we observed is, the lipocalin-2 knockout mice, it exhibited the exacerbated development of arthritis. That means it is the lipocalin-2 is required to control the ongoing inflammation. That's what it is. It gave us an idea. So if you look at this one, wild type, see the disease severity is moderate. Whereas in the lipocalin-2 knockout mice, it is accelerated. The disease is accelerated. So what does it mean? It is the lipocalin-2 is trying to control the inflammation. It is trying to control the inflammation. As I explained in the background, maybe it is trying to repair the damage mediated by the inflammatory disease. So it tells us maybe it is playing a protective role during the development of arthritis. So now, to see what type of cells are infiltrated, what are the cells which is causing this much damage in the lipocalin-2 knockout mice, we carried out the histopathological studies, and what we observed is a significant damage of metacarpal bone and cartilage bone. it significantly degraded the metacarpal and cartilage bone. That leads to the formation of a pannus. That means it is causing the deformities, bone deformities. It is leading to the knockout mice is leading towards the deformities of the bone when compared with the wild type mice. Okay? Having said this, again, the question is, what types of cells are doing this damage? Mainly, it has been shown that KBN serum-induced arthritis model, neutrophils and macrophages are the major culprits. Then we started investigating whether is the neutrophil is infiltrated or macrophage is infiltrated. What we observed, the neutrophil infiltration, how we did? We stained for the Lysix and the DAPI for the nucleus. Wherever we see this polynuclear, the nucleus, that is the typical representation of neutrophils. So we took advantage of this one, the DAPI staining and the Lysix staining, and then we observed under the fluorescent microscope. And what we observed is a significant reduction in infiltration of neutrophils. So neutrophils are not the one who is doing this damage. Then what type of cells? If that is a case, if neutrophils are not coming, because it has been shown that neutrophils are very essential, though it comes and damages the tissue, it is required for later stages, required in the later stages to repair the damaged tissue. Okay? So it is not coming, so that means it tells us the function, adhesion or function of the neutrophil is compromised in the lipocalin-2 knockout mice. Okay? Now, what is the next cells? When we looked into the macrophages, considerably more number of macrophages are accumulated at the joint, where in the damaged area, suggesting that macrophages might be the culprits, which is causing the severe damage. Okay? So, having said this, the take-home message of this presentation is, we are concluding that lipocalin-2 is playing dual role. That means it's playing two roles. In the first skin inflammation model, what we observed is, in the knockout mice, the inflammation is reduced, right? Whereas in the autoimmune arthritis condition, the disease severity is accelerated. So it is a two important roles. That means what it suggests is, this protein, an innate immune protein, which is required for the initiation of the inflammation. First, it acts as a chemoattractant and attracts all the inflammatory cells. Then it eventually, during the later stages, it helps the tissue remodeling cells to correct the damaged tissue. That's what our take-home message. That means the lipocalin-2 is playing a host protective role. So targeting this lipocalin-2 may be a good novel therapeutic strategy in the future. Having said this, at this point, I'm happy to thank my lab members. Dr. R.V. Moola is a postdoctoral fellow in my lab, and Envu is a technician. Austin Russell is a master student. Farhan is work study student. Along with this, I'm thankful to my collaborators at PCOM, Dr. Sharma and Dr. Wang, and at Emory, Dr. Selvaraj, and Penn State University, Dr. Matam. He's the one who gave us the wild type and lipocalin-2 knockout mice. And this study is supported by American Heart Association. Having said this, I will take your questions or suggestions or discussion. I'm sorry. Oh, yeah. We did that. So the lipocalin-2 knockout neutrophils has a defect in adhesion, and it cannot even migrate. When we the other group, I think from Germany, I don't remember where the group is, but the corresponding author is David Weiss. What he did is he conducted a transfer, the migration assay. when you try to do the migration assay, the neutrophils which lacks the lipocalin-2 is not able to migrate, and it's not even able to adhere to the cells, because the infiltration requires the adhesion of the cells, right? So that function is lacking in lipocalin-2 knockout neutrophils. Yes, ma'am. That is an ongoing experiment. That's an ongoing experiment that we are doing now. Recently, it's like almost 6 months or 7 months back, there is one group which published a paper in PNAS where they developed the antagonist for this. But still, how effective it is, it's not yet validated. Okay.
How the lipocalin-2 works for the cells like neutrophils and macrophages? Are there any receptors there, or directly bind to something?
Including T cells, I will say at this time. They expresses the receptor. It is called 243R. That is a specific receptor for lipocalin-2. So what it does is it binds to its receptors, takes the iron inside, and delivers there and comes out. It's recycling. The protein comes, it delivers the, it's almost like a gradient. If the extracellular space is loaded with the iron, lipocalin-2, the secreted lipocalin-2 takes up the iron, binds to its receptor, transports inside the cells, delivers the iron, and comes back. So that's what it does. It is receptor mediated.
So what kind of receptor are they? Are there some GPCR or some?
No. It has a special receptor. It is recently identified. It is called 243R. It is specific for lipocalin.
Thank you.
So it is not like it is characterized like IgG family or transporter family. It's not yet been characterized in that way.
Thank you for a very good presentation. I have a doubt regarding in diabetic condition, the wound healing is delayed because of the infiltration, delayed infiltration of macrophages. So will this help in that condition?
Well, that's what we are trying to address. What exactly it does is it's a proposal that we already submitted. So if you look at the macrophages, there are different subsets, right? M1 macrophages and M2 macrophages. So once the macrophages infiltrates into the site of inflammation or anywhere in the organs, like in pancreas or so, they get differentiated into M1 and M2. So M1 initiates the inflammation, whereas M2 takes care of it. Tissue remodel, inhibits the inflammation. So when we took the macrophages from the lipocalin-2 knockout and we derivatized to the M1 and M2, M2 has a defect in secreting the anti-inflammatory cytokines. So that means the M2 macrophages are not fully functional in lipocalin-2 knockout mice. So there are some studies which is going on with the diabetes also to try to address this. That is the remark.